This is the fourth webinar in a series on the use of AI for federal statistics. And we want to take this opportunity to thank our sponsors, the American Statistical Association, NORC at the University of Chicago, and RTI International. Their funding has allowed us to make this free for all participants. All right, I'm Linda Young, and this webinar is on image analyses, methods, and use cases. I'm Director of Research and Development at the USDA's National Agricultural Statistics Service. If you'll remember, we did for those who were able to attend, we did AI methods for text analyses as part of our second seminar or webinar. And Ben Rogers did a nice job of giving an overview to help set the stage for the speakers. We didn't designate a speaker for that purpose today, so I'll begin with a high-level overview of some basic ideas. First, we need to think about what format the images come in. And the two formats that are the most common are raster and vector. For raster images, a gridded system called a bitmap is used to store colors associated with each grid cell or pixel. For vector images, mathematical formulas are used to capture the shape and colors of polygons, lines, and points within a geometrical framework. You'll see here more info with a link to a website. We'll have this on several slides. These links provide introductory discussions of the concepts on the slide, and you may find them useful for later. Now, for vector images, the AI algorithms for text analysis should be considered. Here we have an example that was taken from Wikipedia illustrating that. And since we've already discussed the methods for text analyses, I won't discuss that further today. Now for raster images, we have the grid, and the pixels are colored for each grid, and the colors are decoded as intensities of red, green, and blue light. So just as we do with a TV set, you have red, green, and blue. The smaller the value, the less intense the light is for that basic color. If it's 0, it's absent. And the larger it is, the more intense it is. Now, the multiband, as for these three colors, images are organized in tensors. And we can think of tensors as n-dimensional matrices. So we have the 3x3 grid, and then we have the three colors. So we have three-dimensional, 2 to get the grid, and another one for the colors. And so we have a three-dimensional tensor. Typically, convolutional neural networks are at the basis of AI models for image analyses. For image processing, we can do a number of different things. We may want to identify features of interest. Say we want to remove clouds and shaded areas on remotely sensed image so we can get better views of the landscape. We might want to identify anomalous images or anomalies within the image. This is used for medical screening of rare conditions or disease. And we've used it as well to detect flooded areas or wildfires from satellites. Now, signal separation is basically separating the signal from the noise. And this is needed when we have a mixture of information in the same pixel. As an example, suppose we have a road in the middle of a pixel when the focus is to determine crop type. We want to remove the noise associated with the presence of the road. One of the useful things for AI is various forms of classification. It could be we want to take a single image and classify it using one output taxonomy. So we might have an image of a flower and want to classify it according to species or its common name. A single image may be classified using several output taxonomies. For example, suppose we have an individual classified and we want to classify that person based on the image according to sex, age, hair color, or any other attribute. Instead, we could have several images and classify those using one output taxonomy. For example, remotely sensed images may be used to identify land cover. Or several images or several pixels within an image may be classified using several output taxonomies. Here we'd have input image being used to classify soil layers as an example at different depths according to different taxonomies, such as permeability classes, soil types, etc. AI segmentation is a set of classification methods designed specifically for images. Segmentation is used to delineate areas of an image and assign them attributes and classes. These are used to retrieve patterns that resemble those seen during training. Now, ChatGPT is in the news, and we've heard many things about its use for text-based analyses. But we have some statistical uses of generative AI methods for images as well. We can be used to impute for areas where information is missing in an image or not directly observable. We can increase the image resolution, which is a form of subpixel analysis. And I think many of us have done this when we've tried to sharpen pictures taken on our phone or by a camera. And disclosure procedures can be developed to privatize, remove, or censor sensitive graphical information. Then there's AI for animated images, which are videos without audio that depict a sequence of images through time. These are often used for displaying temporal statistics, maps across time, or 2D sections across a third dimension, as for medical images. In these and other cases, AI models can be used for change detection. Kasuma will discuss that in her presentation in just a bit. Or screening and surveillance, which is the topic that Scott will discuss. Smoothing, such as interpolation between frames or layers, are useful. Forecasting methods, where we extrapolate nonlinear patterns to obtain in future trends. And this is a use case that Luca will be highlighting. Also, there's backcasting, where we get predictions in the past that are made by going backward in time. Now I'd like to take the opportunity to introduce today's speakers. Scott Lee is our first speaker. He came to CDC in 2014 as an ORISE, maybe or research participant in the National Center for HIV, Viral Hepatitis, STD, and TB Prevention's Division of Tuberculosis Elimination, where he focused primarily on the statistical design of studies for improving TB case identification and diagnosis. In 2015, he moved to the Office of the Director in the Center of Surveillance, Epidemiology, and Laboratory Services, where he joined the Machine Intelligence and Data Science team and worked on a wide variety of projects involving the application of machine learning to problems in public health, surveillance, outbreak responses, forecasting, and diagnostics. In 2023, he completed a 4-month detail to the Coronavirus and Other Respiratory Viruses Division in the National Center for Immunization and Respiratory Diseases. While on detail, Scott served as the acting lead for the Biostatistics, Economics, and Model Unit in the Office of the Director and continued to the development of methods for assessing interference between co-circulating respiratory viral pathogens in pediatric populations. Our second speaker is Lucas Satari, and he is a senior research associate for the National Institute of Statistical Science at NISS, working with the National Agricultural Statistics Service, NASS. He has been involved with the estimation and calibration of the U.S. Census of Agriculture. He worked on modeling livestock, yield, and acreage for major agricultural commodities using various data sources, and he has also developed methodologies for assessing uncertainties. His contribution on the automation of analytical systems has focused on machine learning, artificial intelligence, and high-performance computing. He received his master's in statistics from the Ca' Foscari University of Venice in Italy, and his PhD from the University of Padua, also in Italy. After his PhD, he joined the European Center of Living Technology as a postdoc, researching evolutionary algorithms in AI for 1 year in Venice. Since 2013, he has maintained several packages on the Comprehensive R Archive Network, CRAN, one of which is currently used for production at NASS. Our third and final speaker will be Kusuma Prabhakara. She is a geographer in the Geospatial Reference Data Branch within the Geography Division at the U.S. Census Bureau. She is an imagery lead on the Automated Change Detection Core project, which aims to utilize moderate and high-resolution imagery to focus on areas of detected change and use machine learning algorithms to extract building footprints for use in our intelligence database. Prior to joining the Census Bureau, Kusuma was a graduate research assistant for the USDA's Agricultural Research Service Hydrology and Remote Sensing Lab, working on remote sensing of winter cover crops. This research culminated in a PhD from the Department of Geographical Sciences at the University of Maryland, College Park. Scott, we look forward to your presentation.
Go ahead and share my screen. That looks like it's coming through okay. I hope. Okay. All right. Thanks for the introduction, Linda. So I'm like Linda said, I'm here today to talk about some of the work that we've done in with image analysis at CDC. This is one of kind of a number of use cases that we have going on right now. And this is our project, Harnessing Machine Learning to Eliminate Tuberculosis, or Hamlet. It's a really bad pun, TB or not TB, as is the joke there. But yeah, let's go ahead and dive in. So a quick introduction to what this project is all about. CDC, in collaboration with the Department of State and their overseas partners, is responsible for coordinating the overseas health screenings for immigrants and refugees who are looking to come to the U.S. And so about somewhere between 500 and 600,000 of those get done every year. So it's a lot. And the group that I worked with for this project was CDC's Division of Global Migration Health. So their main missions really are to keep kind of bad stuff from coming into the country, but also to try and keep the populations outside of the country that might come in healthy, just for general reasons. And so they, as part of the overseas health screenings, tuberculosis is one of the main things that the panel physicians look for when they're doing these exams. So there's always a medical history, physical exam, those types of things. But the key point here and the interesting part for this project is that all applicants that were 15 years or older also receive a chest X-ray. And those chest X-rays, I think around 2018, started being transmitted digitally to CDC. So, the agency has a fairly large backlog, basically, of these images, and we weren't quite sure kind of like what to do with them because it's a really big number. And, it's I don't know, it was a really rich data set. So kind of the genesis for this project really was in a very vague sense, thinking about, well, what do we do with all of these digital chest x-rays? And one of the things that I sort of settled on and suggested to the group as a good sort of initial use case for those x-rays would actually be training a machine learning model to kind of do basic diagnostics with the x-rays, really. So, looking for signs of tuberculosis, both specifically in terms of like the individual abnormalities that you might find, like cavities or other types of lesions in the lungs, but also generally just kind of classifying, the x-rays as either abnormal or not. And the main use case we had for this actually isn't where this kind of work tends to go, which is on the clinical side. So looking at using, AI to kind of boost, clinical capacity in resource limited settings, because the Overseas Health Screening Program is relatively well resourced to begin with. So really our use case is actually internal quality control. DGMH is interested in kind of seeing how all of these panel sites overseas are doing in terms of catching the stuff that they're supposed to catch and, not flagging too many false positive x-rays for having problems. And so our goal really was to develop an algorithm that could replicate what the clinicians were doing in the field, but use it internally to effectively give us a really rough look at what their work might be like. Okay, so the methods for this, we tried to cobble together good data sets for this. So the kind of background caveat is that all of the images that I originally had to work with were from people who had already been admitted to the US successfully. And so typically those images don't have many abnormalities on them. And so a large part of what we did at the beginning of the project was just trying to get together a fairly representative data set where we had lots of like healthy images, which is great, but we also needed abnormal ones, and specifically ones that were consistent with signs of tuberculosis. So we got these data sources together. eMedical is the Department of State's system. I think it's actually run and maintained by the immigration authorities in Australia. But that combined with the International Organization for Migration's refugee health system kind of gave us most of the images that we used for the project. So that was on the order of about 150,000 that we had for training. And the proportions there were about 50%, 15% of those were abnormal and 9% were abnormal, suggestive of TB. But to kind of put together the validation and test sets, which are kind of like the most important, because the whole reason we're building this model is to effectively have a double check against what the panel physicians are doing in the field. Then using those fields labeled images as testing data doesn't really make sense. So we worked with a couple of external partners that see relatively high TB caseloads, and they also have their own QC kind of controls put in place. So we worked with them to put together validation and testing data sets. And for that, we also tried to upsample the number of images that had signs of active tuberculosis, so we could have better representation for that. So just a quick overview of the data set and its characteristics. So again, about 15% were abnormal across all of the data sets, and then 9% were abnormal suggested with TB. The second box here is really just a breakdown of all the different kinds of abnormalities that we saw. And the reason I point this out here is really just to show how rare some of these were. Like, we're looking at hilar mediastinal adenopathies as being like 0.2%, right, of the total images. So this is kind of I guess, telegraphing one of the results that didn't turn out very well for us, which was trying to predict these abnormalities specifically themselves instead of just the general classification for the images. But the final note here is that, I mentioned this just briefly before, images from people who actually went on to test positive for tuberculosis were really fairly rare in the data set because of the relative level of health of the populations that we were working with. And so really we only had about around 1% of those across all of the training data sets sort of met those criteria, which limited what we were able to do with the project. I talk to people about this project, and often they ask, well, can you predict TB? And it's like, well, no, because one, that wasn't the goal of the project, is really to look for abnormalities in the x-rays, but two, we just didn't have the data to do that kind of training. And then so for testing also, I figured it's always a good idea to test against as many external data sets as you can find to try and get a sense of generalizability. And so these were the external data sets we used. Chest X-ray 14 from NIH is really, really common in the literature. And then so are these other two TB specific data sets. They're very, very small and they've been around for a while, but all of the abnormal images in these data sets came from people who actually had like either, like lab confirmed TB or molecularly diagnosed TB. So those are really valuable there. And then the last data set was also relatively large and had a good blend of images from different abnormalities. So we tested against those as well. Just a brief note on the model architecture. I won't really get into this, but for this project, we use SufficientNet v2s, which I think by now is probably like completely out of date, but it worked really well. And the reason I have the graphic on the right is really to show that it's super, super efficient for training and for inference, which is the main reason I picked it, just to speed up kind of those early project iterations where you're trying to get code to work. But it also leaves a lot of performance kind of on the table. So some of the bigger models, right, on that graph are going to do much, much better, at least on ImageNet, which is what the graph is showing. And so it's kind of this compromise. So we probably have more performance we can squeeze out of the data using a stronger model. But mostly it was pretty standard stuff. I added some custom layers in the model for like pooling and dropout and the final classification. And then also importantly added some image augmentation layers at the input level of the model. So during training time, those are going to do all these random permutations to the images, the X-rays as they're coming into the model, effectively increasing the training data set size and the variability of like image quality really that the model sees to make it more robust against outliers in the future. And so just a note for like feds is kind of interesting. How do you get this work done, right? I mean, we've got way more resources for doing deep learning now than we did 5 years ago, but really all of this work was done on just a standard scientific workstation that CDC gave me. All the code was done in Python. Most of it was fairly custom, but we did use pretty normal like libraries for this kind of stuff like TensorFlow and Keras. Code was designed to be run as sequentially executed scripts, which was sort of my attempt to make it something akin to a workflow that could be rerun when TGMH had new images that came in. And then, sorry, EDAV here is a CDC specific acronym. Should have taken it out, but basically we got all of these scripts ported into our cloud environment so that now this can be run in a little bit more automated fashion without me having to do it manually. Quick info on our training and testing procedures for the model itself. It was really simple. Like the data set was really big for training, and the images and the labels were actually pretty clean. So it only took about 1 to 2 passes through the data for AUC to begin or decreasing on the validation data, which was our early stopping metric that we used. For decision thresholds for like turning those predicted class probabilities into actual like class predictions, like abnormal or not, those were all chosen on the validation data. And then, yeah, for statistical inference, I know this is kind of the statistics community. It's not really interesting. It just bootstrapped a bunch of stuff. So that's sort of what the main analysis was at the end. And I did some kind of ad hoc, maybe a little fuzzy reweighting during the bootstrap replicates to try and account for the higher than normal prevalence of abnormal images that we had in the test data because we intentionally oversampled those. All right, so results. How did the model do? Another big table of numbers, but we'll try to focus on the important bits. So, I've got our data sets that we use running down the left side. Hamlet is our internal data set that we gathered. But you can see across all of the data sets, the AUCs are really pretty high. On the Shenzhen and Montgomery data sets, those are really small. And again, there's pretty good class separation between, the healthy folks and the not healthy folks there. So it's typical for people who are working with those to post really high AUCs. But even on the data set from Vietnam, which is relatively new, still doing pretty good at predicting whether images are abnormal and suggestive of TB. So that was the first thing we're kind of excited to see. Another kind of niche metric, I guess, is specificity at 90% sensitivity. And then sort of the reverse, which is sensitivity at 70% specificity. One of those, and I don't remember which, and I'm sorry for that, is used by the WHO as basically a minimum threshold that a diagnostic test needs to meet, a screening test needs to meet for being used in the community to screen for TB. We weren't looking specifically at TB disease. It was really about whether the image was abnormal, the X-ray itself. So this doesn't really apply, but I pointed out just to show that the models are still, our model is still doing pretty well against that standard if we were to use it. And then, yeah, let's see. The final note from this slide really is that these are the results just for predicting generally whether images were abnormal. So they didn't need to be suggestive of TB. Any abnormality would be fine. And we're still seeing pretty good results here. That 92 AUC is well below state of the art on chest X-ray 8 and 14, which I think is around 96 the last time I checked. But 92 is still pretty solid. So, and we weren't really training the model for that purpose specifically. That was kind of an after effect. So those are pretty good AUCs overall. This is what I was alluding to earlier about having the model try to identify the specific abnormalities. You can see here that while the AUC is on the right column, oh, you're like, hey, 93, that looks great. 92, 86, 96, those aren't bad. Because some of these things are so rare, the PPV is actually like really bad, like unusably bad. So for at least for the meantime, we're sort of scrapping this part of it and really just focusing on the binary classification moving forward. I also, just because I thought it would be cool, worked in some code to the project to generate heat maps. This is just using openly available methods for doing this kind of attribution work with images. But they produce these pretty cool results. And so this is a figure from our preprint that I have, where it's, it's just what we got here, like 5 X-rays, right? And then different heat map methods showing kind of where the abnormal spots are predicted to be based on the model. And the last one on the far right is really cool because it sort of does this really nice segmentation based on the predicted probability of abnormality. And what you can end up doing is actually just masking out areas of the X-ray that the model thinks aren't important at all and really drawing focus to the areas that it thinks are important. And so eventually, if we think about moving this, this project sort of into the clinical domain, even if it's just experimentally, I think having this kind of interpretability will be pretty helpful. So conclusion really quickly, for all of the fancy stuff, I think that went into the project, and as challenging as it was for me to put together, because this was the first time I'd really worked with image data, basically all I did was just wrote a bunch of Python code that turns a batch of DICOM files, which are like the container files that have the X-rays, just turns a batch of those into a spreadsheet that kind of looks like this, where, we've got the images on the rows and then just the predicted probabilities of abnormality across all of the different categories. So that's kind of the end result here. And the only reason I show this is because sometimes it's nice just to see, like, right, you put all this really cool stuff together, what does it actually generate? And this is basically what it looks like. And so I think for federal folks who are on the line and kind of interested in this, one of the questions that I get asked a lot is like, well, how did you like kind of bring all this together? Really, one of the key things was collaboration. All of the technical work I did myself, all the programming, but there was so much else that needed to go into this project in order to make it happen. And so having really strong partnerships, both inside the agency and outside the agency, was really critical, especially for getting us data that we need. And so we have these results. What's next for the project? So the first thing is that really we have solid evidence that the models that we trained actually do work really well. And they're all in line with results that we see published by groups from like Google, who are kind of working in the space too. We're getting similar metrics with them on the data sets that we tested. So that's really encouraging, just from a, hey, this actually works perspective. And so the first thing that we're actively using this for now is basically setting up those internal quality control procedures. So when DGMH gets a really big batch of X-rays, say like quarterly, you can run the model on those, get the model's predictions, and then sort of crosswalk those with what the actual, clinical readings were, and get a sense for if there are really big discrepancies between those two, which there shouldn't be, then that might be something that we need to look further into. So really triggering a manual review at, say, a panel site specific level. The future, there's lots of other future use cases that are kind of interesting, especially like predicting TB disease, maybe with multimodal models that take the X-ray and clinical and demographic information into account. But mainly for right now, we're just really happy to have something that can do QC at scale on these images. So some quick acknowledgements. Hamlet team members, couldn't have done it without them. Importantly, we also worked with a lot of these panel sites overseas that were really, really great to work with, and were really responsive to all of our kind of pestering data requests. And yeah, that's it. So I'll turn it over to the next speaker. Also, apologies for talking super fast. Just trying to get in under the time limit, so I appreciate it.
Good afternoon. Today I'm going to present something related to the predictions for cropland data layer. This is going to be a showcase for how we get from images to statistics. So how to explore the predictive methodology within an estimation cycle from a statistical perspective. We have, for the past 3 years or more, We have started to look at the estimation that occur in June. So every year at NISS, we have a publication for planted acreage, and this is like a state level report of so we have estimates for a different set of crops, and these are usually produced by combining several sources of information that includes survey data, but also we have administrative data from the Farm Service Agency, which are typically used as ground truth in combination with the remotely sensing images. However, most of the FSA data are usually not available until July, but when they're available for later months, they do not cover all the fields. So predictive approaches are usually needed, especially in June, to address concerns on the missing data from the survey. And so if we want to do some sort of imputation, the estimation as well, because these are additional information that we can use not just from the survey, but we can merge other data to get improved estimates. And the other questions that we have addressed, or we're even still addressing, is related to the uncertainty quantification, how we can estimate standard error when we combine all this information. So currently, this is like a very brief diagram that shows how the data flow for the estimation is occurring at NISS. So once we have the remote sensing images, we produce like some sort of data fusion methods that we use to obtain land cover classification. This land cover classification is that fused with other data from the survey. And once this fusion is finalized, we can produce estimate for acreage. So if we look at the evolution of the remote sensing images, here I have produced like the median for the true colors, which are the red, blue, and green. We also use other spectrum bands that are like the near infrared and shortwave lengths, but these are not represented here. So once we look at the evolution of these patterns across times, we can see that in April, nothing was really planted. In May, we start to have something that's planting. But then in June, we have some fields where already the vegetation is thriving, but other fields are planted in June. Once we reach July and August, all fields are planting, the vegetation is grown and keeps growing. Then once we reach September, we have the first harvesting, and October, the final part of the harvest. By looking at this evolution of the colors of these spectral bands, we are able to detect through machine learning the type of crop that has been planted on certain fields. By doing so, we are able to create the cropland data layer. This is primarily developed to inform the planted acreage estimation. And as I said, we use the remote sensing images and the machine learning methods to classify the land cover and produce this output. This output is produced every year for the 48 conterminous states since 2008. And every year we published about 9 billion pixels at 30-meter resolutions. And this provides information for over 100 crops cultivated in the US. But if you remember, in April, we do not have all the information from these, phenological curve, where these patterns can inform us on the crop that has been planted, because in April, nothing has been planted yet. So we started to look into crop rotation patterns and how we can use the information from previous CDLs that we have published with some correction from the administrative data that at the end of the year, of every year, they are fully available at that point for some of the fields. And once we merge these two data sources, we are able to track through time crop rotation patterns, and through a modeling approach, we are able to make some prediction. But once we start to go into the growing season, and after some of the crops have been planted, we can start to use also remote sensing together with the crop rotation patterns from the past CDLs and the administrative data. So this means that to produce a predictive CDL, we need to process 9 billion pixels for every year that we use as an input. So we tend to use at least 5 years of data in input to this model. But in research, we are also exploring for options about over 8 years, from 8 to 10 years. So these models that here have depicted into 3 neural networks, but especially the first one and the second one are pre-trained on separate sources of data to produce a predictive distribution. However, once we have these specialized networks, we can perform transfer learning. So we use their pre-trained set of weights, and then we merge the output as an input for a third neural network that it is used to make the final prediction of the distribution that we want to use for the predictions and uncertainty estimation. Once we perform the training, we do noise injection. This noise injection is obtained through dropout layers usually. And we make, through this approach, the model more robust to outliers and also to measurement error. Once we obtain the predictive distribution through the entropy, we can assess the uncertainty of those predictions. In this case, for us, the maximum entropy, if from a uniform choice where the selection of the categorical prediction becomes completely random, the entropy for that randomness, which is the maximum in that case, is going to be 2.4. However, in here, for the case of Illinois, the entropy, which measured the ambiguity for the selection of the category, goes from 0 to 0.5, which is way lower than 2.4, which is the maximum theoretical. At this point, once we zoom in on specific areas, we see most of the green patterns are on average entropy. There is some uncertainty on the selection of the crop prediction here. The blue areas are related to urban areas and bodies of water. And fields that are yellow and brown are more uncertain. And in this case, they're more related to fields that have been inundated during 2019. And because there was a disruption in the pattern for the crop rotations, the model was able to capture an anomaly within the pattern for making the prediction, but also we were able to capture the uncertainty of that anomaly within our pattern and being able to display it through the maps. Also, when we analyze the relationship between entropy and accuracy in the classification, we realize that the fields that have highlighted here with this red ellipses, they tend to have a higher entropy, and those higher entropy fields are associated with less accurate classification results. In fact, on the left, some of those fields have been either classified from corn and soybeans with a lot of uncertainty. But once we start to use more remote sensing images throughout the year, those uncertainties tend to diminish and we make better prediction. So once we have our classifications and we conduct the surveys based on an area frame. So the area frame consists on statistical units that are actually delineated segments, and these segments that are depicted here in red on the figure have been sampled. Some of these segments that have been sampled, we get through data collection. And once we finish the data collection, some of these segments have missing information. Currently at NISS, we go through manual imputation, and we have explored ways to either automate that or simplify it or increase the revision of this manual imputation process. through a semi-automatic imputation method that applies our AI predictions that either coming from neural networks, and we have also other machine learning approaches as well that comes into place here. We also evaluated the quality of our predictions, and this quality is based on the ground truth data that is coming from the FSA, which is usually collected post the June area survey, which is after July. Meaning the June area survey happens in June, but the administrative data are available after July. So once we have the fields that have this missing information, we have these boundaries that delineate the areas that we have to make some predictions. So what we do, we overlay these boundaries on our predictions, and then we summarize the area, the acreage, for specific crops of interest. In this case, if we look at the fields that we here call tracts, for example, those that have a cropland indicator of 1, we are able to summarize the acreage for corn and soybeans in a specific case, but also we have other crops of interest that we can summarize from these predictions. Once the data are tabulated, we assess the accuracy, and what we saw that for the respondents, our prediction was 81.6 in R-square, so very correlated with the output. But once we have used the no response cases after a second stage of interview, so we have a follow-up where we still collect some data through interview, we were able to determine that our prediction model was 0.952 in R-square, and it was even more correlated with acreage from the survey. But because there is still some variability, we then developed a model to determine the variance of these predictions as well. So the initial thoughts that we had, it was to determine that the planted acreage has some physical bounds, so it cannot be smaller than 0, and it cannot be larger than the area of the field. So once we developed the model, we accounted for heteroscedasticity, where the input variable that we have used to determine the variance for our imputed values are based on tabulated entropies, which occurs with a similar system as we saw before, but rather than using the categorical crop prediction, we use the entropies. We also determine the differences between different prediction models as well. And the total acreage of the sample units as well is considered within this modeling technique. The approach is based on generalized additive models for location, scale, and shape functional parameters. And these are developed for a specific chosen distribution. Once we look at the residuals in terms of true error from the administrative data and the behavior of our standard error for those units, we were able to determine that 50% of these points were below 1 sigma, the blue line. About 95% of the points here are below the line of 2 sigma, the green line. And about more than 99% of the data points in this graphic were below the red line, which is the 3 sigma threshold that we are used to for to consider in statistics. Then the other aspect that we considered was how we blend this information from the crop predictions, the imputed values, and the survey data that we have for national estimates. As I said here in this slide, we have a model-based, there are estimates produced through a model at the state level, but we have better models at the state level that we used. These are just preliminary results, and once we aggregate them from the state level to the national level, we compare with the NISS official statistics. Once we use the survey only, we get some results that are good. But once we start to look at the model blend, most of them are even better. In a special case for 2019, which was a year where we had heavy rainfalls, where the planting patterns were disrupted in several states. We still have good results for corn, but for soybeans, we were not able to achieve a relative error below 5%. This means that this model had to be further improved by considering other variables and aspects that we haven't considered yet. Overall, these are preliminary results, as I said, and they are showing a particular application of AI and machine learning models on images that are finding promising potential for statistical application. The main AI research areas at NISS that focus on image analysis are on crop prediction, classification, computational efficiency, for which transfer learning is part of it, imputational reliability, and uncertainty quantification. Also, we look at different models, not just AI and machine learning, but Bayesian models as well for blending sources of information. We have also been part of the early adoption and testing for the IT infrastructure as a team. These technologies allowed us to make analysis for the data at a national level, and this has been very beneficial for our team because allowed us to test this technology even for other divisions within NISS. These are some references. Part of this work has been done in collaboration with my colleagues at NISS as well, with other colleagues at NISS as well. And thank you so much.
Okay. My name's Kasuma Prabhakara. I'm a geographer with the Census Bureau, and I'm going to talk about satellite-based detection and delineation of built environment features today. First, let me give some background on the evolution of what we call in-field canvassing. So traditionally, in-field canvassing has ensured that the Census Bureau can refine the master address file, or MAF, in advance of the decennial census, typically in the years ending in 8 and 9. So this canvassing was designed to validate the address list and add, change, or delete addresses as necessary in the MAF. However, the way we have conducted address canvassing has evolved over the last 3 censuses. In 2010, 100% of in-field address canvassing was performed through paper-based operations using books of printed address registers and printed maps at the assignment area level of geography. In 2010, 100% of in-field canvassing was performed using handheld devices that captured address updates in GPS and relative coordinates. In 2020, there was a major shift in methodology. 100% of the addresses in the address frame were reviewed during in-office address canvassing as opposed to in-field, and 35% of the addresses identified for in-field address canvassing as areas of possible coverage issues. So to go into a little bit more of what we did in 2020, essentially, as I said before, a key mission of our division is to update and maintain the MAF. Historically, this was undertaken by field canvassers and through data sharing partnerships with tribal, state, local, and federal partners. So in 2020, we built a block tracking database. It was based off of census blocks, which are the smallest geographic area for which the census tabulates and collects decennial data. We conducted a complete review of all blocks in the nation, some of which were reviewed multiple times to evaluate ongoing change. And in total, we conducted more than 13 million block reviews. We identified growth, decline, and built-out areas, and built-out areas were flagged to minimize the need for field canvassing. This effort required nearly 150 full-time staff over a 4-year period. Some of the results of this is that we reduced the number of housing units requiring a field visit by 65%, reduced the field staff for canvassing from 150,000 in 2010 to 32,000 in 2020. And it also resulted in a cost avoidance of approximately $674 million versus going to all housing units in the field. So what do we want to do? What we want to do is automate the manual change detection methods we used in 2020. We want to automate in-office updates to the master address file, or MAF, and also the topologically integrated geographic coding and referencing system called Tiger, which is essentially shapefiles. We want to increase the quality, coverage, and spatial accuracy of MAF Tiger and increase efficiency of field operations and optimize resource allocation. Our objectives are to develop and execute automated methods, models, and implement information storage systems that enable identification of change on the landscape. We plan to do this by running an ongoing moderate resolution automated change detection process on the nation every 6 months. So our plan for the moderate resolution change detection is essentially our focus is identifying land that has been cleared for potential construction or construction that has recently occurred. So this will allow us to focus on areas with coverage issues and where updates may be needed to our MAF Tiger system. These data, along with other resources, can help us validate and automate updates. The way that we're looking for areas of change from vegetated to non-vegetated using moderate resolution is kind of an ensemble approach. A few of the different methods we're using, including calculate the difference between Sentinel-2 recent images and long-term normal NDVI, or artificial surface indexes, to detect change of 10 meters. And again, we're using Sentinel-2 imagery. It's the source because it has a 5-day revisit, multiple bands of data per pixel, and it's relatively manageable size in comparison to higher resolution imagery. Another way we're looking for the change from vegetated to non-vegetated is using a NASA product called Opera's Dist Alert, and this can find persistent disturbance and be used for validation. Opera stands for Observational Products for End Users from Remote Sensing Analysis, in case anybody was wondering. We're also using Dynamic World to detect areas of change between built classifications, and then our own time series analysis against training areas that have been created within the census. And again, these models are going to be used together to have higher confidence in the change areas that we've detected. So once we've detected change polygons using our moderate detection or moderate resolution detection, we acquire high resolution imagery based on the Military Grid Reference System, or MGRS grid cells, and we get high resolution imagery for the grid cells containing change polygons that we got from the moderate resolution change detection. Currently, we're going to use the Building Footprint Extraction USA and the Multi-Task Road Extractor deep learning models for extraction of buildings and roads, respectively. Instance segmentation will occur using masked region-based convoluted neural networks and faster RNN architecture. Staff will review the 1 kilometer cells identified using the moderate resolution change detection. Additionally, building footprints were generated in these grid cells from the RCNN model using high resolution imagery. Staff will review the footprints generated for accuracy and completeness and create footprints that may have been missed by our model. The edited data is then transformed into training data and used to improve the model's accuracy and robustness for future runs. After we have our building footprints, we can intersect and integrate information and use them in our intelligence database. So now let's dive a little deeper into how we actually do the moderate level change detection. The yellow outline you see is a 1 kilometer grid cell from the MGRS, and that's our unit of work in this model. The satellite images in these, the satellite images are Sentinel-2 from May 25th, 2023. And the blocks of color that you see on the right-hand side are from the NASA Opera product that I mentioned earlier. And in red, you can see where it indicates persistent change from a vegetative state to a potential cleared construction site longer than 30 days. So using this data, we can pinpoint areas of potential growth and move on to the next step. So the next step is we download high-resolution imagery from the National Geospatial Intelligence Agency Maxar Global Enhanced GeoInt Delivery API. You can see the yellow highlighted grid is the same as on the previous slide, but the high-res imagery is much clearer. So on the left, you can see the area before the change was detected, and on the right, you can see the detailed changes based on more current high-res imagery. We added another layer of data in this step, building footprints from USA Structures or Microsoft, and we use that for update and validation. We're also extracting our old building footprints using the Esri Deep Learning Kit object detection software. And this data helps us track detailed changes over time. So on the right, you can see structures highlighted in different colors. So the orange structures we were able to detect March 19th, 2023. The green structures were captured March 30th, 2023, and the blue structures were captured May 23rd, 2023. So as a vote of confidence in our approach for using machine learning and imagery to detect change on the landscape, it should be noted that many of the automated change detection methods being employed by the census are also becoming common practice among other federal agencies in the private sector. So with that, I will close. And if you have any questions, this is me and my colleague, and we would be happy to answer them. Thank you.
I want to thank all three of our speakers. You did an outstanding job. I see that some are putting questions in the Q&A. Please use the Q&A instead of the chat. And I will start answering those. I want to give you a little bit of time. So I'm going to start with the first question. And that are, you know, we take a lot of precautions with our data when we, so we have different disclosure limitation methods. We have differential privacy, for example. But what are some of the harms or risks that are unique to image-based models as opposed to, say, text or traditional machine learning models? Do we take as much care with the images as we do with our other collected data? And if not, does that matter? Nothing like a showstopper to start with, is there? Scott, you look like you have some thoughts. You're using individual level X-rays, so.
Yeah, no. You know, I mean, obviously for this project, we tried to get the best performance possible because it's, you know, it's I mean, even in our setting where it's not clinical, it's stateside, it's just, you know, internal QC, it's still fairly consequential, right? If we have a model that kind of goes off the rails and starts, you know, pegging specific panel sites as doing not a very good job, and it's, you know, wrong, then we, you know, send a bunch of people on a goose chase they don't need to go on, and it looks bad. It just looks bad for everybody. So yeah, I mean, I think the consideration for this project, for me personally, as the developer that I maybe haven't followed in past projects, was really trying to get, you know, the best performance that I possibly could from classification, given the technical constraints that I had. Like, you know, the workstation I had could only fit a certain size model on it, so I couldn't try one of the bigger ones. But yeah, and I don't think I mentioned this in my slides, but we were also careful to make sure that none of our images had any burned in patient information on them. So in those DICOM container files, there's always metadata about, you know, the X-ray machine itself, but also a lot of information about the applicants that were looking to come to the US. And so I used optical character recognition to sort of scan all, you know, all of the data that we had for the project, and then just omit anything that, you know, had more than, I don't know, 2 words on it or something like that, just as there's been extra measure for trying to protect privacy. But yeah, no specific considerations. I think just, you know, a sense of that it was important, and we wanted to try to do the best that we could.
Right. Taking the name off of it or any other personal identifier is a big one, though. I have to agree. Luca or Kasuma, did you want to add to that at all?
Well, yep. So for the disclosure when we could use generative AI is when there might be some sensitive area in that we do not want people to see directly. So in that case, it could be replaced by a pattern that has been observed somewhere else to cover those specific sensitive areas. You know, in agriculture, we don't do that, but because we want to see the field. So actually, we would like to do the opposite. But there are some situations for sensitive areas, you know, where you have maybe a nuclear plants or something like that that you would like to hide. That could be helpful.
Mhm. Kasuma, did you have anything to add?
I was just going to say, I mean, you know, with census, we always need to be really careful about privacy and privacy issues. So a lot of the data that we're doing the modeling on is, you know, publicly available. But then when it goes into our intelligence database is kind of when we have the, you know, privacy issues. And we're very careful about it, though. I think that's important with imagery analysis.
I think it is for all of us. And one of the challenges, I think, is that we can go to Google Maps and see so much of the stuff that we're working with anyway. And it's actually combining it with other data, as you point out, that I think really leads to some challenges. We now have several questions. And Scott, you've been diligent about answering some of these, but I'd like to go ahead and highlight one or two. For the internal QC, how did the models perform at local individual country? And how would you handle countries where model wasn't working, or maybe there are local QC issues?
Yeah, so I'll try to answer the parts that I can answer. You know, the big caveat here is that I'm not sort of an SME in that division, sort of responsible for the day-to-day programmatic, you know, workflows. I was very much sort of the outside guy that got called in to help with the programming parts of it. But as far as I understand, their initial sort of QC test of the model beyond what we did for the paper, right? So they have a really enormous backlog of images, like I mentioned. So there's plenty of extra sort of informal testing data that we can use. I think that's still ongoing. The idea there was really to get getting the model's predictions on the data is really quick, relatively. But you know, clearly to understand whether it's doing what we want it to, we need to have manual review. And so as far as I understand, our contract radiologist is still kind of looking through those and trying to identify, you know, where the mismatches were and what might have caused them. Because I think sometimes, you know, I mean, often with models like this, it's huge, it's super powerful, and so it might be queuing into things that aren't necessarily clinically relevant, right, to make these determinations. So I think that that's still under progress. And as far as, you know, model quality across sites, I think I answered this in the question, but I would expect it to stay mostly the same. Clearly, the like background rates of TB in places will be different, but I mean, at least what I saw sort of informally looking at performance across sites, it tended to be pretty similar just because, you know, chest x-rays are chest x-rays and there's certain minimum quality standards that our program requires. So image quality itself isn't really a driving factor there. And then, yeah, I mean, if there are sort of issues that crop up, I actually don't know how those would be handled. I'm assuming very carefully and responsibly. So, but, you know, hopefully we don't have those pop up, I think would be the ideal.
Yeah, it would be ideal. So Luca, on slide 7, can you talk more about the type of noise dropout layers injected and how you quantified the increase in robustness of the model?
Yes, definitely. So the dropout uses like a Bernoulli distribution with a probability, and the dropout probability is then used for a normalization. So technically what happened is we use the input and we multiply by this Bernoulli, so it's going to be a random 0, 1. So wherever there is 0, the output is not going to be used into the next calculations. When we have a 1, is going to be used. However, once it is going to be used, it needs to be reprocessed because the expectation of the input information is not going to be equivalent to the expectation of the output. And so by doing this reprocessing is like saying, okay, The model needs to see the whole information, but at times during training, some of that information is hidden. And once we do multiple iteration of that process, which is random, is like saying, okay, the model is not going to be allowed to see the whole information at once. And just by doing that, you increase robustness because if not all the information is available, the model try to get the best of what has been feeding. And there are other ways, however, to inject noise without using dropouts layers. But in that case, we're going to have noise that is randomly generated from other distributions that is going to be added to the input values. And, you know, once we move forward from the input towards the output, that noise propagates. And the model needs to account for that. And once we produce the results, we remove this randomness from the model. And so from the input to the output, the model is gonna produce more reliable outputs. And more or less, that's the mechanism behind.
All right, thank you. If you have a follow-up question, just stick it in the Q&A. So Kasuma, you have a couple of follow-ups by the same individual. During your process of identifying new structures, can you differentiate between the commercial and residential structures, one, and will you please elaborate how you use the acquired data?
Sure. So we do later on, we use parcel data in conjunction with our building footprints to determine the type, whether they're commercial or residential. And as far as how we use the acquired data, we kind of mash it together with some other data sets that we have to give a more complete view of the different buildings. So we have some accessory data sets, or ancillary data sets, I should say.
All right, thank you. Now, before I ask each of you another question, do you have any questions you want to ask of each other? I haven't really given you a chance to do that yet. There's a pause, but so you can think about it. I'll come back to you in just a moment. So Scott, were the data imbalanced when training, testing, and validating the model? If so, did you consider using precision recall AUC?
Yeah, so the data were really pretty imbalanced, right? I mean, the overall across our, all our training, validation, and test sets, only 9% of the images were abnormal and suggestive of TB, which was our primary sort of like category that we were after in classification. I didn't do anything during training to adjust for that, and it seemed to perform fairly well. In terms of reporting the metrics, I mean, and I alluded to this, or just said this explicitly in my response to the answer. I mean, the main reason I highlight AUC is because that's what everyone else does, whether it makes a lot of sense or not. So, that's the metric that kind of gets floated around in the literature in the space. And so mostly I focus on AUC for parity with existing work to show that, hey, like, so for example, on the chest X-ray 8/14 data sets, how does our number stack up to what other groups are getting? But again, and I apologize for not highlighting this in the presentation. I think the Zoom speaker box was actually covering up the right side of the screen for me. But the metric we actually care the most about for this project is kind of a strange one for thinking about like using AI for chest radiography, but it's really how well does the model predict the number of abnormal images in a batch, so that when DGMH gets a big chunk of these things in, we can really quickly eyeball, okay, the model says that 10% of these are abnormal, but it's reported that 20% are abnormal or something like that. So for that metric, it doesn't even matter how good the individual level classifications are. It's really just, at that batch level. And so that's really the metric that we actually do focus on. And for that, the model did fairly well. I think on our test set, it was off by around 2%, I think, of the actual count. which is small enough, I think, to be sort of useful for our purposes.
All right, thank you. Luca, have you considered applying OpenET or something similar to gain information about water usage and add that data to your model?
We had initial thoughts about water usage and other type of data sets related to water, like soil moisture and things like that. But we haven't used it yet. It's something we have to explore, though.
Do you have that data available?
I need to check with my colleagues.
Yeah, okay. It just seems to me that might be hard to get track of, to find. All right, Kasuma. Let's see, I had one here. How do you identify areas where population growth may have occurred without significant detectable change in satellite imagery?
So yeah, this kind of goes back similar to other questions. We have the ancillary data sets that we use. So we can look on like the delivery sequence file from USPS or some other sources to see if things have changed, you know, that we haven't necessarily been able to capture through change detection.
All right. So, and this one will go for all of you. Are there any image data sets that are underutilized within your agency? So thinking of things like satellite imagery, clinical X-rays, scan documents. In other words, what is the potential for additional applications of AI to images within your agencies?
I'll take a first stab at that just because I think with my use case, it's fairly straightforward. So as far as I know, this project was the first one to look at, really applying AI to medical imaging data and then, tying that to a specific public health use case. There aren't a ton of other programs at CDC that would benefit from chest radiography specifically. I mean, we do have a domestic TB program, and we've talked with them about like working together on future projects and things like that. But there are lots of other, I think, open, like really big open data sets that could be useful. So CDC has a really big and strong infectious disease pathology group. So there's been a ton of work in that space specifically outside of public health, but just in the world. And so that's a space I kind of see us maybe being able to move into and try to capture some of, I mean, the great thing about a lot of these models is that really what they're doing from a diagnostic sense is capturing clinical expertise and making it redistributable, right, in a way that doesn't require the person to be sitting there looking at a film or looking through a microscope. So yeah, I think infectious disease histopathology is probably something worth looking at, and there's been lots of great work on that. And yeah, other than that, I'm actually not really sure. Oh, right, yeah, dermatology, right? So there's also been a ton of work on applying AI to just pictures of molds, for lack of a better, more technical description. And I know that there are several groups at CDC, I think, who have kind of batted around applications with those kinds of data, either for screening or whatever. So I think I'd like to at least see us explore those temporarily. I don't really know where they would go, but it would be interesting, I think, to take a look.
I can say with our work that, one thing we've considered looking at is like radar, because you can really get textures and things from radar. We just don't really, at the moment, have the kind of computing and processing needs that we need to kind of do that. But so there are some underutilized data sets that, are out there that could be used.
Yeah, I mean, we have also data sets from images that maybe are not from NISS, but are produced by other agencies that we do not utilize. But we might have ideas on how to merge them and bring them in for future users as well. Yep.
Do you have an example of one that you think?
Well, for example, right now we're not using weather data on temperature. There could be some patterns that may shift the decision farmers made during planting. So if there is like a warmer weather early in the season, then it's possible that they are going to plant earlier. Then a combination between flooding and, well, flooding, that was 2018, but there are like heavy precipitation, things like that. We have explored some uses, but we haven't used them for production. So that could be something we still need to look at.
Mhm. So, you each had some techniques that you used, but what are some techniques within image recognition that hold promise for the mission of federal agencies? So just kind of envisioning things. So for example, localization, segmentation, OCR, image retrieval, and what kind of applications could you envision for those techniques? Any ideas, suggestions?
So for us, I know, this isn't maybe the part of the agency where I work in, but a lot of our recent data modernization efforts have focused on kind of improving our ability to both, receive and to send data to and from jurisdictions, so state and local health departments, hospital systems, those sorts of things. And I know optical character recognition is kind of a big part of that, because as we always hear, a lot of places are like still submitting case reports by fax and things like that. So I know OCR is kind of factoring into how the folks in the agency who are working on that modernization effort are thinking about it. From my perspective, I think there's like with this Hamlet project, I don't want to say it was low-hanging fruit, but it kind of was. It's like there's this existing task, it's very clear, we have all the work done outside of public health, there's code that I can use to put the model together. And so I think a lot of, from my perspective, a lot of the things that would be worth pursuing are really pretty straightforward, and most of them probably are just going to fall under some kind of image classification. And then also building out internal tools, so like vector databases to help diagnosticians kind of search images they've seen before, not seen before, those types of tools. But yeah, from a practical standpoint, I feel like classification can get you really pretty far, with what you're able to do, and kind of conversely, the amount of effort that it takes to put it together.
Yeah, I would think, especially in the medical field, that's primarily what you want to do. You want to classify something as healthy or unhealthy and things of that nature. So I can certainly understand that. Kasuma, do you have some thoughts?
Sure. So I think that image segmentation with the goal of deriving footprints using CNN and change detection, which are what we're working on. I mean, I could see that being useful across lots of different agencies and have actually like multiple applications beyond what we're doing. And I think that's why it's kind of catching on so much.
Right. So one of the things, it's a little different, but change detection, as we have witnessed greater effects of climate change, the crops are tending to move north and west in the United States to some extent, the plantings of crops. So being able to detect that change is a little different from the application that you mentioned, but being able to detect the change and model that change over time would be of interest, and also of great importance to as far as food security goes. So that, just to build on what you talked about. Luca?
Yeah. What I think we are not using yet, but it's gonna be a possibility, is the integration between raster images from satellites and current in-house segments. There are polygons, so they're more like vector type of images. So blending the two aspects from different data sets, where we have the raster images and the vector images for certain areas. We recently published the CSBs, which is a crop sequence boundaries. Those are delineated fields from the CDL, but they provide the contiguous areas that could be used for further improving these models. So that could be an example of how to merge two different type of images as well into a single model, trying to make predictions. The other would be even multimodal type of approaches where, yes, we have within this general model for images is going to be part of these other bigger model where we have other data sets that are coming in format that are like tabular or text even, depends on the situation, but it would be more an integrated approach for the estimation.
All right, thank you. There's an interesting question that's just been posed in Q&A. Do any of you have examples of areas where there might be data in other agencies that can improve your model? For example, Kasuma mentioned tax assessor information is useful for census. Have you been able to dig into information the Department of Treasury collects?
I'm not super familiar with that side of it, but maybe one of my colleagues on the call is more familiar with it. I don't know. I'm not too familiar with that.
Looks like Elvis may have something.
No, I'm not too familiar actually myself. So,
All right. We can, I can think of quite a few data now, whether it helps the models or not. There are a lot of data in other agencies that I would value. So it's just whether or not they help these types of models, right? Scott?
Yeah, I mean, for us, obviously, like, NIH, there's a few folks in the National Library of Medicine who have, been working in this space for like more than a decade already. And I know NIH is going to have lots of good sort of rich, chest radiography data, and then also the VA, just because of their, extensive network. And they're actually pretty, I think I don't know if they're the most advanced, but I've always been impressed with the sort of the ML and AI work that I've seen coming out of the VA. So yeah, those are two groups that I'd be really interested in talking with. I will say it's kind of like, we're at the point with this project where we're sort of evaluating, sort of the initial effort, and does it really work? Does it not work? Because it's one thing to train up a model and use it to make a table of results for a paper, but it's another thing to actually see, as part of day-to-day operations, how does that actually help? But I think once we get through that phase, we'll definitely start thinking about, phase 1B or phase 2 or something like that, where we start seriously looking at augmenting our initial data set, with other X-rays, especially to try and, improve our ability to pick up some of those really rare abnormalities. I don't know that we'll ever get to the point where like the model's working well enough to really do that and have it be useful, but there's only one way to find out, right? Get lots of data and try and make the model better. So yeah.
Modeling is an evolutionary process, I find. Luca, any thoughts?
Yeah, like agencies that are external to USDA, I would think about NOAA and NASA. We're already using Landsat 8 and 9 for the crop data layer. And that's produced by NASA. Lately, NASA is also producing, well, I mean, lately, a while now. He's producing like a harmonized Landsat and Sentinel-2. The Sentinel-2 is remote sensing data from the European Space Agency. So these are like harmonized. They're like providing more coverage throughout the nation. But those are already in use. There might be other data sets that I'm not aware though. From NASA. From NOAA, we have all those data sets that we haven't explored yet. And I'm definitely not well knowledgeable, but it's something that we still need to further investigate, I guess. Regarding USDA agencies, I don't think we have used yet RMA data, Risk Management Agency. But we are gonna have some collaboration soon that we are going to start using them for other type of projects that are image related. But I can't think about anyone else further than these ones, of course.
All right, thank you. Do any of you have a final word before we draw this to a close? If not, I want to thank you, all three of you, again. You did a very wonderful job of illustrating applications and uses of AI on particular use cases, and that always brings some concreteness to the big ideas. Thank you so much for participating today. And those of you who came, we really appreciate your participation. Thank you.